Good morning, dear students. Welcome to Genesis. Let us quickly discuss some important questions from the chapter Absorption by Roots. Now, in this question, they have given you a diagram, and just by looking at the diagram, we can make out this is a diagram of a root hair in the root. And they have asked few questions related to the diagram. Now, let's read question number 1. Name the plant parts indicated 1 to 4. So, they have asked you to label 1, 2, 3 and 4. 2 definitely is soil particles. 1 is root hair. 4, this is cortex, and third is xylem. So the parts are root hair, soil, xylem and cortex. These are the 4 parts. Which process enables the passage of water from soil to the root hair? The process by which water can move into the root hair cell is called osmosis. If they would have asked you name the process by which solutes enter into the root hair, then the answer would be active transport. Third, name the pressure responsible for the movement of water in the direction indicated by the arrow. Which pressure is responsible for forcing the water in this direction? Now we can very clearly say that the water is entering by cell to cell osmosis in the root hair cell. Now the pressure which is generated due to the cell to cell osmosis within the root is known as root pressure. Last question, name the phenomena that happens in plant due to excess of this pressure. So they are asking you which is that phenomena that happens in plant due to excess of root pressure. We all know that due to excess of root pressure in small plants, the water is given out from the margins of the leaf vein through pores called hydrothores, and that process is known as guttation. So this is the phenomena that we will see in plants due to excess of root pressure. Now let's discuss question number 2. Question number 2, we have a setup where we are showing this is a U-tube where the 2 limbs of the U-tube are separated by a semi-permeable membrane. And there is sucrose solution present in both the limbs. However, there is a difference. In limb A, there is 10% of sucrose solution, whereas in limb B, it is 5%, which clearly means that the concentration of sugar molecule is more in limb A compared to limb B. The first question is, what phenomena is shown in the diagram? Since there is semi-permeable membrane separating the 2, so there will be exchange across the semi-permeable membrane, which happens in the case of osmosis. Because this is not a living cell where they can spend ATP to perform active transport. That could also have happened. But here, since it's an artificial setup and it is just separated by semi-permeable membrane, so if you're asked what will happen here, then definitely what will happen is osmosis. Second, which limb of the U-tube contains more concentrated sucrose solution? Clearly, we can see that limb A has more number of sugar molecules compared to B. If you ever get confused, just count the number of sugar molecule. Whichever side has more sugar molecule is more concentrated. So the limb A has or contains more sucrose compared to limb B. Why is the membrane separating semi-permeable? It is semi-permeable because it will allow only solvent to pass. So this membrane only allows solvent to pass. That is why it is a semi-permeable membrane. Which limb of the U-tube is comparable to the root hair of plant? This is an excellent question. They are asking you out of the limb A and B, which one imitates a root hair cell? So basically, we have to have this knowledge that the root hair cell has a cell sap which is highly concentrated compared to the water which is present in the soil. So why is it more concentrated? Because it has more salts in it. So out of these 2, if I ask you which is more concentrated, then it is obviously A which is more concentrated. So this is imitating a root hair cell. So the tube A will be the answer to this question. Last, redraw the diagram to show the result. What will happen after some time? We know it is a semi-permeable membrane. What will happen is osmosis, which will only allow solvent molecule to pass through it. So the solvent molecule will move from a higher concentration gradient to lower concentration gradient. Now here the quantity of solvent molecule is more compared to in limb A. So the movement will happen from limb B to limb A of water molecules. So what will we see? After some time, the level of water in tube A will rise. So when you are redrawing the diagram, what you basically do is you just change the level of water and that's it. You are going to show that the water molecules, they moved from limb B to limb A, thereby resulting a rise in the level of water. Let's do next question. In question number 3, they have given a setup, and we are already quite familiar to this setup, where they are showing there is sugar solution in the thistle funnel, there is water present in the beaker, and the mouth of the thistle funnel is sealed with a semi-permeable membrane. Now the first question is, name the process being studied. The process that is being studied here is osmosis. Why? Because there is a semi-permeable membrane separating the two conditions. One, which is a solution of sugar, another is plain water. Second, what changes would you observe in the thistle funnel after 10 minutes? After 10 minutes, we are going to see a rise in the level of the water in the thistle funnel due to the movement of water molecule from the beaker into the thistle funnel. Or you can also say due to the process of endo-osmosis, which will happen from the beaker to the thistle funnel. So there will be a rise in the level of water. Next is sugar solution a hypertonic or hypotonic? The sugar solution is hypertonic in comparison to the water that is present in the beaker, because it has more solute compared to that in the beaker. Last, number 4, explain why much salt is added to pickles. Now pickling is a process where we try to preserve vegetables and fruits for a longer span of time. And how do we do it? By killing bacterial infestation. And how can that be done? By adding more sugar or more salt. This will make the condition hypertonic, and in hypertonic condition the bacterial cell will die by the phenomena of plasmolysis. That means they are going to lose their water due to the presence of the hypertonic external environment, and they die out of it. So we are going to meet again in my next video with some more questions.